Your heart just stopped. For a fraction of a second between beats, you were technically dead. And then, without any conscious input from you, your heart contracted again, pushing a fresh surge of oxygen-rich blood through your arteries, sustaining your brain, your muscles, your very existence. This miracle repeated itself while you read this paragraph, perhaps 70 times or more. But have you ever wondered what makes your heart beat at all? What mysterious force orchestrates this perpetual rhythm? The answer lies not in some mystical energy, but in one of the most extraordinary evolutionary adaptations in the history of life on Earth. A biological innovation so elegant yet counterintuitive that it borders on the magical. Imagine for a moment that your existence depends on the continuous operation of an ancient electrical system. Now imagine that this system must function flawlessly through sleeping and waking, through calm and crisis, through every second of every day for decades, without a single reboot or moment of maintenance. This is not science fiction. This is the reality of the electrochemical engine that powers your heart, a system so remarkable that its failure means the end of you. What if I told you that the most critical component of this life-sustaining system, the part that keeps your heart beating right now, evolved from something entirely different? That through a twist of evolutionary fate, an ancient genetic accident repurposed primitive cellular machinery into the sophisticated pacemaker that now governs your heartbeat? This is the story of that strange evolutionary trick. A tale that begins not with humans, not even with vertebrates, but in the ancient primordial oceans nearly 2 billion years ago, when life was still figuring out what it meant to be multicellular. It is a story about how evolution tinkers, repurposes, and transforms, creating complexity from simplicity, order from chaos, and ultimately, the rhythm that sustains us all. In the next 15 minutes, we will journey from single cells to complex bodies, from ancient oceans to modern medical laboratories, uncovering the improbable series of adaptations that led to the remarkable system that keeps you alive right now, with each and every heartbeat. The year was 1882, and in the quiet laboratory at the University of Cambridge, a physiologist named Walter Gaskell was puzzling over the strange behavior of a turtle heart. Unlike mammalian hearts, which beat so quickly their movements blur into a continuous motion, the cold-blooded turtle's heart beats slowly enough to observe in detail. Gaskell noticed something peculiar. The heartbeat always originated in the same location, a small region where the veins enter the heart, before spreading in a wave-like manner through the rest of the organ. This observation might seem trivial today, but in 1882 it was revolutionary. For centuries, physicians and anatomists had debated what makes the heart beat. Aristotle believed the heart contained an inner fire. Medieval scholars thought it housed the soul. Even by the 19th century, many scientists believed that nerves from the brain commanded the heart's rhythm, like a puppeteer controlling a marionette. Gaskell's observations suggested something radical. The heart possessed its own intrinsic timing system, independent of the brain. The heartbeat originated within the heart itself. Around the same time, in 1883, German physiologist Wilhelm, his junior, was peering through his microscope at thin slices of embryonic hearts. He noticed a strange bundle of specialized tissue connecting the atria, the upper chambers, to the ventricles, the lower chambers. This structure, now called the bundle of his, was unlike normal heart muscle or nerve tissue. Its purpose remained elusive, but his suspected it might be important for coordinating the heart's contractions. The mystery deepened in 1893 when another physiologist, George Ralph Mines, performed a clever experiment. He carefully separated a frog's heart into pieces and discovered that each piece could still contract rhythmically on its own, albeit at different rates. The piece containing the junction where veins entered the heart, the sinus venosus, always beat the fastest, setting the pace for the intact heart. These scattered observations were like pieces of a puzzle, waiting for someone to assemble them into a coherent picture. That someone was Arthur Keith, a Scottish anatomist, who along with his colleague Martin Flack made a breakthrough discovery in 1907. While examining the heart of a mole, they identified a small, specialized region of tissue at the junction of the right atrium and superior vena cava. precisely where the heartbeat originated. They called it the sinoatrial node, though today we know it simply as the heart's natural pacemaker. It is, I believe, Keith wrote in his journal, the remnant of the primitive cardiac tube, which has retained the power of rhythmical contraction which the rest of the heart has almost entirely lost. Keith's intuition was remarkable. This tiny node, barely the size of a grain of rice, was indeed an evolutionary remnant, a living fossil within our bodies. But the true story of its origins was far more ancient and extraordinary than even Keith could have imagined. The significance of these discoveries was monumental. They revealed that the heart beats not because the brain tells it to, but because specialized cells within the heart itself generate electrical impulses automatically and rhythmically. Your heart will continue to beat even if all nerve connections to the brain are severed, a fact that makes heart transplantation possible. But this raised a deeper question. How did such a system evolve? What possible advantage could there be in developing tissue that spontaneously generates electrical impulses? To understand this, we need to journey back in time, to an era long before hearts existed, to the very dawn of multicellular life. The story begins not with hearts at all, but with something far more fundamental. The cell membrane and its remarkable property of maintaining different concentrations of ions inside and outside the cell. This creates an electrical potential difference, a voltage, across the membrane. All living cells maintain this voltage, from bacteria to brain cells, and it is one of life's most ancient and conserved features. This voltage is not static. It can change when ion channels, specialized proteins embedded in the cell membrane, open or close, allowing ions to flow across the membrane. When this happens, the voltage changes rapidly, creating what scientists call an action potential, essentially a brief electrical pulse. In single-celled organisms and many primitive multicellular creatures, these electrical properties serve various purposes: detecting prey, avoiding predators, or coordinating basic movements. But as animals became more complex during the Cambrian explosion over 500 million years ago, these cellular electrical properties were repurposed for something new: communication between distant parts of an increasingly complex body. And here we encounter one of evolution's most remarkable principles. Adaptation, the process by which a trait evolved for one purpose is repurposed for an entirely different function. The classic example is feathers, which evolved for insulation in dinosaurs before being repurposed for flight in birds. In our case, the cellular machinery that generated electrical signals for basic functions in simple organisms was about to be repurposed for something far more sophisticated: the coordination of a muscular pump we now call a heart. But to understand how this happened, we need to look at creatures that exist in the evolutionary space between organisms without hearts and those with fully formed ones. We need to examine the transitional forms that reveal how this extraordinary adaptation emerged. Imagine the prehistoric oceans about 600 million years ago. The animal kingdom was still in its infancy. No vertebrates yet swam these waters, only simple invertebrates like jellyfish, primitive worms, and early mollusks. These creatures had no hearts as we would recognize them, but they were not without circulation systems entirely. Consider the jellyfish, one of Earth's oldest surviving animal lineages. These gelatinous creatures have no central heart, yet they move water and nutrients through their bodies using a network of canals lined with tiny hair-like structures called cilia. These cilia beat rhythmically, creating currents that distribute nutrients throughout the jellyfish's body. What makes these cilia beat in a coordinated fashion? The answer reveals the first clue in our evolutionary mystery: specialized cells called pacemaker cells that generate rhythmic electrical signals, causing nearby cilia to beat in synchrony. Dr. Zoe Jenkins, a marine biologist at the Scripps Institution of Oceanography, has spent decades studying these primitive coordination systems. What is fascinating about jellyfish is that they show us a kind of protocardiac system, she explains. They have these specialized pacemaker cells that coordinate muscular contractions for swimming, but also help circulate their internal fluids. It is not a heart, but you can see the fundamental cellular machinery that would later be adapted for cardiac function. These pacemaker cells in jellyfish contain ion channels that automatically open and close, generating rhythmic electrical impulses without any input from a brain or nervous system. This property, automaticity, is the same property that allows your heart to beat without conscious control. It is a beautiful example of deep homology. Jenkins continues, referring to the concept that similar structures can exist in very different organisms because they evolved from shared ancestral genetic material. The ion channels that create automatic rhythmic firing in jellyfish pacemaker cells are molecularly related to those in the human sinoatrial node. Same basic machinery, repurposed for a more complex function. As we move forward in evolutionary time to about 550 million years ago, we encounter more complex invertebrates like flatworms. These creatures have a slightly more sophisticated circulation system, a network of fluid-filled spaces called a gastrovascular cavity that distributes nutrients throughout their flat bodies. Dr. Howard Simmons, an evolutionary developmental biologist at Yale University, explains, flatworms represent an important transitional form. They have specialized muscle cells that contract rhythmically to move fluid through their bodies. These contractions are coordinated by primitive pacemaker cells that, again, show this property of automaticity, the ability to generate electrical impulses without external stimulation. What is remarkable about these flatworm pacemaker cells is that they express many of the same genes that will later be found in vertebrate heart tissue. Genes with names like NKX2.5, GATA factors, and T-box transcription factors, the same genetic toolkit that builds the human heart, are already being expressed in these primitive circulation systems. When we look at the genetics, Simmons notes, we see that the blueprint for the heart was being assembled long before the heart itself existed. Evolution was tinkering with these genetic circuits, gradually assembling the components that would eventually come together to form a proper cardiac system. The next major advance came with the evolution of segmented worms like earthworms about 500 million years ago. These creatures possess multiple hearts, really just specialized blood vessels with muscular walls that contract to push blood through the body. Each of these contractile vessels has its own pacemaker cells that generate rhythmic contractions. But there is something different about these pacemaker cells compared to those in jellyfish or flatworms. They have become more specialized, more autonomous, and more reliable. They express a more complete set of the genes we associate with heart development in vertebrates. What we see in segmented worms, explains Simmons, is the beginning of a hierarchical organization. Some pacemaker regions are dominant over others, setting the overall rhythm. This is the precursor to the dominance of the sinoatrial node in the human heart, the ability of one group of pacemaker cells to override others and coordinate the entire system. This pattern of increasingly sophisticated pacemaker systems continues as we move through the evolutionary tree to mollusks like squid and octopuses, which have relatively advanced hearts with distinct chambers and powerful muscles. Their hearts contain well-defined pacemaker regions that set the rhythm for cardiac contractions. But the most significant leap occurred with the evolution of the earliest vertebrates about 520 million years ago. The fossil record reveals creatures like Haecoichthyes, a tiny, fish-like organism from the Cambrian period that had a notochord, a primitive spinal cord, and likely possessed a simple tubular heart. This primitive vertebrate heart represented a quantum leap in circulatory efficiency. Instead of multiple contractile vessels or simple pulsing tubes, it was a centralized pump with specialized tissue for generating and conducting electrical signals. The genetic toolkit that had been assembled piecemeal in invertebrates was now deployed to build a dedicated organ for circulation. The vertebrate innovation was centralization and specialization, says Dr. Maria Rodriguez, a paleocardiology expert at the Natural History Museum in London. Instead of distributed pumping systems, they evolved a single, powerful pump with dedicated pacemaker tissue. This allowed for higher metabolic rates, greater size, and ultimately, the active lifestyles of vertebrates. As vertebrates continued to evolve, this simple tubular heart became more complex. Fish developed a two-chambered heart with one atrium and one ventricle. Amphibians evolved a three-chambered heart with two atria and one ventricle, allowing for some separation of oxygenated and deoxygenated blood. Reptiles began the transition to a four-chambered heart, with some species like crocodilians achieving complete separation of the pulmonary and systemic circulation. Birds and mammals, independently, evolved fully four-chambered hearts with complete separation of oxygenated and deoxygenated blood, enabling the high-energy lifestyles these animal groups are known for. Throughout this entire evolutionary sequence, spanning hundreds of millions of years, the fundamental property of cardiac automaticity remained constant. From the primitive pacemaker cells of jellyfish to the sophisticated sinoatrial node of mammals, the ability of specialized cells to generate rhythmic electrical activity without external input was preserved and refined. But what exactly makes these pacemaker cells tick? What molecular mechanism allows them to generate electrical impulses automatically, without any conscious control? The answer to this question would not begin to emerge until the mid-20th century, with the development of techniques to measure the electrical properties of individual cells. The year was 1952, and at the University of Cambridge, physiologists Alan Hodgkin and Andrew Huxley were making history. Using the giant axon of the squid, a nerve fiber so large it can be seen with the naked eye, they measured the electrical currents that flow across cell membranes during an action potential. Their groundbreaking work, which later earned them a Nobel Prize, revealed the fundamental mechanisms of how nerve cells generate electrical signals. But something was different about heart cells, particularly those in the pacemaker regions. Unlike nerve cells, which fire only when stimulated, cardiac pacemaker cells fire spontaneously, setting the heart's rhythm. It would take several more decades before scientists fully understood the molecular basis of this cardiac automaticity. In the 1970s and 1980s, researchers discovered a family of ion channels unique to heart cells called funny channels or HCN channels, hyperpolarization-activated cyclic nucleotide-gated channels. These HCN channels have a property that seems almost, funny. They open when the cell membrane voltage becomes more negative, contrary to most ion channels. This unusual property creates a slow, steady influx of positive ions into pacemaker cells, gradually raising the voltage until other ion channels open, triggering an action potential and a heartbeat. Dr. Robert McKinnon, a cellular electrophysiologist at Johns Hopkins University, explains, "What makes cardiac pacemaker cells so special is this built-in clock mechanism. The HCN channels ensure that after each heartbeat, the cell automatically begins preparing for the next one. It is like a metronome that never needs winding." But here is where the story takes a fascinating turn. When scientists examined the genetic sequence of these HCN channels, they discovered something remarkable. They are ancient. Versions of these channels exist not just in all vertebrates, but in many invertebrates as well, including some single-celled organisms. These channels did not evolve specifically for the heart, McKinnon explains. They were repurposed. In primitive organisms, similar channels help regulate basic cellular functions like volume control or environmental sensing. Evolution co-opted them for a completely new role in cardiac pacemaking. This repurposing principle extends beyond just ion channels. The entire genetic program that builds and regulates the heart has deep evolutionary roots. The transcription factors that control heart development, genes with names like NKX2.5, GATA4, and TBX5, have counterparts in animals that evolved long before hearts existed. For example, the gene NKX2.5, which is crucial for heart development in humans, belongs to an ancient family of genes found even in sea anemones, which have no hearts. In these primitive creatures, the gene helps organize the body plan and develop the digestive system. Only later was it recruited for cardiac development in more complex animals. This pattern of repurposing is a recurring theme in the evolution of complex biological systems. Evolution rarely invents new components from scratch. Instead, it tinkers with existing parts, adapting them for new functions. Your heart's pacemaker is built from genetic and molecular components that existed hundreds of millions of years before hearts themselves evolved. But the story of cardiac evolution becomes even more intriguing when we consider the neural crest, a group of embryonic cells unique to vertebrates that contribute to many structures, including parts of the heart. During embryonic development in vertebrates, neural crest cells migrate from the developing spinal cord to various parts of the body, including the heart. These cells contribute to the cardiac outflow tracts, the aortic arch arteries, and, critically, parts of the cardiac conduction system, including tissues surrounding the sinoatrial node. The involvement of neural crest cells in heart development represents a vertebrate innovation, explains Dr. Nicole Anderson, a developmental biologist at Stanford University. It allowed for greater complexity and sophistication in cardiac structure and function. The neural crest contribution may have been crucial for the evolution of the advanced conduction systems we see in vertebrates. This neural crest contribution creates an interesting paradox. While the fundamental property of cardiac automaticity evolved from ancient cellular mechanisms predating hearts themselves, the sophisticated conduction system that carries these electrical impulses throughout the heart is a relatively recent vertebrate innovation. It is like evolution built a modern electrical grid around an ancient power source, Anderson notes. The basic generator, the pacemaker cells with their funny channels, has roots going back almost to the dawn of multicellular life. But the distribution network, the specialized conduction system, is a more recent development that allowed for the coordination of increasingly complex hearts. This evolutionary history explains many of the heart's peculiarities. For instance, the sinoatrial node, your heart's primary pacemaker, is not located at the center of the heart as one might expect for a coordinating structure. Instead, it sits at the junction of the right atrium and the superior vena cava, precisely where the venous entry point to the primitive tubular heart would have been in early vertebrates. The sinoatrial node is literally a living fossil, says Dr. Thomas Ziegler, a cardiologist at Massachusetts General Hospital. Its location makes perfect sense when you understand that it evolved from the pacemaker region of the primitive linear heart tube of early vertebrates. As the heart folded and developed chambers during evolution, the original pacemaker region maintained its dominance, even though its location ended up being somewhat off-center in the final four-chamber design. This evolutionary heritage also explains why the heart can beat outside the body, why heart transplants work, and why certain cardiac arrhythmias occur. The heart's fundamental timing system is autonomous because it evolved from cellular mechanisms that predated complex nervous systems. It does not need the brain to tell it to beat. It has been beating on its own for hundreds of millions of years. But this autonomy comes with trade-offs. Because the heart's pacemaker evolved long before complex brains, the integration between these two systems is sometimes imperfect. Stress, anxiety, and other psychological states can disrupt the heart's rhythm through complex interactions between the nervous system and cardiac pacemaker cells. The strange evolutionary trick that keeps you alive, then, is not just the existence of pacemaker cells themselves, but the delicate integration of this ancient cellular machinery with newer regulatory systems, a marriage of old and new, of autonomic and conscious, of steadfast rhythm and adaptive response. In the pre-dawn darkness of a hospital room, a monitor beeps steadily, marking the rhythm of a patient's heart. The patient, 67-year-old Harold Sanderson, lies unconscious, his chest rising and falling with mechanical precision as a ventilator breathes for him. 24 hours earlier, Harold suffered a massive heart attack that severely damaged his heart's electrical conduction system. Dr. Elizabeth Chun checks his vital signs, then turns to Harold's daughter. "The damage to your father's heart is extensive," she explains gently. "The natural pacemaker, what we call the sinoatrial node, has been severely compromised. Without intervention, his heart cannot maintain a rhythm strong enough to sustain him." "So what happens now?" the daughter asks, her voice trembling. Dr. Chun points to a small device on the nearby tray. "This is an electronic pacemaker. We will implant it this morning to take over the function of his damaged natural pacemaker." It will detect when his heart rate falls below a certain threshold and provide electrical impulses to maintain a healthy rhythm. This scene plays out thousands of times daily in hospitals worldwide. Artificial pacemakers have become routine medical technology, saving countless lives. But their very existence highlights something profound about our evolutionary history. When the ancient biological system fails, we must replace it with technology that mimics its function. Modern electronic pacemakers are marvels of biomedical engineering, but they are essentially sophisticated mimics of a natural system that evolved over hundreds of millions of years. They generate electrical impulses at regular intervals, just as the sinoatrial node does, but they lack the subtle adaptability and responsiveness of the biological original. Even our most advanced pacemakers are crude approximations of nature's design, explains Dr. Ziegler. The natural pacemaker adjusts not just to physical demands but integrates information from hormones, neurotransmitters, temperature, and dozens of other variables. It is a system of exquisite complexity and responsiveness. This complexity becomes even more apparent when we examine how the heart's pacemaker system develops in the human embryo. By the 4th week of gestation, the embryonic heart has already begun to beat. This occurs before the nervous system has formed connections to the heart, before lungs exist, and while the embryo is still smaller than a grain of rice. How does the heart know to start beating at this precise developmental stage? The answer lies in the intricate genetic program that guides cardiac development, a program with deep evolutionary roots. As the embryonic heart tube forms, certain cells within it begin to express the genes for HCN channels and other components necessary for automaticity. These cells, located at what will become the sinoatrial node, begin generating spontaneous electrical impulses even before the heart has fully formed. It is a beautiful example of how development recapitulates evolution, notes Dr. Anderson. The embryonic heart begins as a simple tube, similar to the hearts of primitive vertebrates. Pacemaker activity begins at one end of this tube, just as it did in our evolutionary ancestors. Then, as the tube folds and forms chambers, this pacemaker region becomes the sinoatrial node. This developmental sequence reflects the heart's evolutionary history, but it also reveals something profound about the nature of biological timekeeping. The heartbeat is one of the body's most fundamental rhythms, a clock that must start ticking early in development and continue uninterrupted throughout life. But the heart is not the only clock in the body. Circadian rhythms regulate our sleep-wake cycles, hormonal fluctuations follow monthly patterns, and even individual cells have molecular clocks that regulate their activities. All these timekeeping mechanisms share common evolutionary origins. Dr. Michael Hastings, a chronobiologist at the Medical Research Council Laboratory of Molecular Biology in Cambridge, explains, "The ability to measure and respond to time is one of life's most fundamental properties. From cyanobacteria to humans, organisms have evolved mechanisms to synchronize their activities with environmental cycles and to coordinate internal processes. The molecular components that drive these various biological clocks often overlap. For instance, some of the same ion channels involved in cardiac pacemaking also play roles in the brain's circadian clock. The genes that regulate these channels have been conserved across vast evolutionary distances, suggesting that biological timekeeping is one of life's most essential functions. This shared evolutionary heritage explains why disruptions to one biological rhythm often affect others. Shift workers whose circadian rhythms are chronically disturbed, for example, show higher rates of cardiovascular disease. Stress can affect both sleep patterns and heart rate variability. The body's various clocks do not operate in isolation but as an interconnected system with ancient evolutionary roots. The heart's pacemaker, then, is just one component of a complex, multilevel timekeeping system that coordinates our physiology across multiple scales, from cellular to organismal. This system evolved not all at once, but through a series of adaptations spanning billions of years, from the first rhythmic cellular processes in primordial bacteria to the sophisticated integration of neural, hormonal, and cardiac rhythms in modern humans. When we really understand the evolutionary history of cardiac pacemaking, says Dr. Simmons, we see that it is not just about keeping the heart beating. It is about the evolution of biological time itself, the ability of living systems to create and maintain rhythm. This capacity is so fundamental that it precedes not just hearts, but even multicellularity. It is woven into the very fabric of life. This perspective transforms how we think about cardiac disease. Arrhythmias, abnormal heart rhythms, are not simply mechanical failures of the heart but disruptions to an ancient biological timekeeping system. Treating them effectively requires understanding not just the heart in isolation but its place within the body's interconnected rhythmic networks. Modern medicine is beginning to incorporate this evolutionary perspective into treatment approaches. Chronotherapy, timing medical interventions to align with the body's natural rhythms, has shown promise for improving outcomes in various conditions, including cardiovascular disease. Similarly, vagal nerve stimulation and other techniques that modify the nervous system's influence on the heart can help restore healthy cardiac rhythms without directly interfering with the heart itself. For Harold Sanderson, the patient awaiting a pacemaker implantation, this evolutionary context might seem academic. What matters to him is that medical technology can replace his damaged natural pacemaker and allow his heart to continue beating. But the very possibility of this intervention rests on centuries of scientific inquiry that revealed the evolutionary trick keeping us all alive: the autonomous rhythmicity of cardiac pacemaker cells. As Dr. Chun prepares for Harold's surgery, she carries with her not just modern medical knowledge but the accumulated understanding of how evolution shaped the human heart. She knows that the device she will implant is merely substituting for a system with origins reaching back to the dawn of animal life, a system so fundamental that its disruption threatens life itself. Let us pause for a moment to consider the sheer improbability of your existence. Not just the astronomical odds against your specific genetic makeup, but the long chain of evolutionary innovations that made human life possible at all. Among these innovations, few are as fundamental as the development of automatic cardiac pacemaking, the strange trick that keeps your heart beating from before your birth until the moment of your death. This trick, this evolutionary adaptation, did not arise fully formed. It emerged gradually, through a process of tinkering and repurposing that spans more than 500 million years. What began as simple ion channels regulating basic cellular functions in primitive organisms was slowly modified, elaborated, and integrated into increasingly complex systems, culminating in the sophisticated pacemaker network of the human heart. The four-chambered heart that powers your circulation is a relatively recent evolutionary innovation, appearing only in the last 300 million years with the emergence of advanced reptiles, birds, and mammals. But the fundamental property that makes this heartbeat automaticity is ancient, with roots reaching back to the earliest multicellular animals and perhaps beyond. This evolutionary history is written not just in fossils but in your very DNA. The genes that build and regulate your heart's pacemaker system have counterparts in creatures as distant from us as jellyfish and sea anemones. When scientists knock out the gene NKX 2.5 in mice, the animals fail to develop proper hearts. But this same gene has relatives in creatures that have no hearts at all. What is most humbling about this research, reflects Dr. Jenkins, is recognizing that the boundaries between organisms are not as sharp as we once thought. The genetic toolkit that builds a human heart shares significant components with the toolkit that organizes a sea anemone's digestive system or coordinates a jellyfish's swimming contractions. We are all variations on ancient themes. This shared genetic heritage reveals a profound truth about evolution. It does not work from blueprints, but by modifying existing structures. Your heart's pacemaker is not designed from scratch, but assembled from components that originally evolved for other purposes. This process, which scientists call adaptation, is a central principle of evolutionary innovation. The implications of this evolutionary perspective extend far beyond scientific curiosity. They inform how we understand and treat cardiac disease, how we develop artificial pacemakers, and even how we think about the integration of technology with biology. Consider the field of tissue engineering, where scientists are working to grow functional cardiac tissue in the laboratory. This research relies heavily on understanding the developmental cues that guide embryonic cells to become pacemaker cells. By recapitulating aspects of evolution and development, researchers hope to create biological pacemakers that could someday replace electronic devices. Dr. Laura Wilkinson, a biomedical engineer at the University of California, San Francisco, explains, "Nature has solved the problem of creating self-regulating cardiac tissue through hundreds of millions of years of evolution. Rather than trying to engineer completely novel solutions, we are increasingly looking to understand and mimic these evolved systems." Wilkinson's team has successfully generated pacemaker-like cells from stem cells by activating the same genetic pathways that direct pacemaker development in the embryo. These engineered cells can spontaneously generate electrical impulses similar to those of natural pacemaker cells, offering hope for biological alternatives to electronic pacemakers. What is fascinating, Wilkinson notes, is that the genetic program we activate to create these pacemaker cells is ancient. We are essentially tapping into evolved developmental pathways that long predate humans, or even hearts as we know them. This approach, looking to evolution for inspiration, extends beyond cardiac medicine to other fields as well. The principles of biological timekeeping that evolved in cardiac pacemaker systems have parallels in the regulation of sleep cycles, hormonal rhythms, and even cellular metabolism. Understanding these shared evolutionary origins helps scientists develop more holistic approaches to health and disease. But perhaps the most profound implication of the heart's evolutionary history is philosophical. The autonomous nature of cardiac pacemaking, its independence from conscious control, highlights a fundamental truth about human existence. We are not fully self-contained, rational agents in complete control of our bodies. We are, instead, complex ecosystems of evolved systems, many operating outside our awareness or control. Your conscious mind, that sense of you reading these words, does not tell your heart to beat. It never has throughout your entire life. Your heart beats because of an ancient cellular mechanism inherited from ancestors so distant they would be unrecognizable to you. Every second of your existence depends on biological systems with deep evolutionary roots. Systems that operate autonomously, reliably, and typically without your awareness. There is something humbling about this realization, reflects Dr. Simmons. The conscious self that we identify with so strongly is just one component of our biology, and often not the most crucial one for survival. Our lives depend on ancient, autonomous systems that operate below the threshold of consciousness. This perspective challenges the common metaphor of the brain as the body's command center. While the brain does regulate many bodily functions, systems like cardiac pacemaking operate with significant autonomy. The relationship between brain and heart is less hierarchical than collaborative, a partnership that evolved over millions of years. This partnership is reflected in the rich two-way communication between heart and brain. The heart sends more neural signals to the brain than the brain sends to the heart. These cardiac signals influence emotional processing, cognitive function, and even consciousness itself. Your heartbeat shapes your emotional state just as your emotional state influences your heartbeat. Dr. Rachel Thompson, a neurocardiology researcher at Oxford University, explains, "We have traditionally viewed the brain as the master regulator of the body, but the reality is more complex. The heart contains an intrinsic nervous system, sometimes called the heart-brain, with over 40,000 neurons that process information and make decisions without input from the brain. This intrinsic cardiac nervous system interacts with the heart's pacemaker cells, allowing for complex regulation of cardiac function in response to changing conditions. It represents yet another layer of the evolutionary trick that keeps us alive. Not just automatic pacemaking, but sophisticated semi-autonomous regulation of that pacemaking. The implications of this heart-brain relationship extend beyond physiology to our understanding of emotions, consciousness, and even cultural conceptions of the self. Across human cultures and throughout history, the heart has been associated with emotions, courage, and essential aspects of identity. While we now know that the brain is the seat of consciousness, these cultural associations may reflect an intuitive recognition of the heart's importance not just for physical survival but for emotional experience. The ancient Egyptians believed the heart, not the brain, was the center of intelligence and emotion. In their conception of the afterlife, the heart was weighed against a feather to determine one's moral worth. The brain, meanwhile, was discarded during mummification as relatively unimportant. While we now understand that the brain is responsible for cognition, the Egyptian intuition about the heart's importance was not entirely misplaced. The heart's rhythm influences emotional states through complex feedback loops involving the vagus nerve, the amygdala, and other brain structures involved in emotional processing. Heart rate variability, subtle variations in the timing between heartbeats, is now recognized as an important indicator of emotional regulation capacity and stress resilience. Low heart rate variability is associated with anxiety, depression, and poor emotional regulation, while high heart rate variability correlates with better stress coping and emotional flexibility. The heart's rhythm is not just keeping you alive in a physical sense, explains Dr. Thompson. It is intimately involved in your emotional life, your stress responses, and even your capacity for social connection. The quality of your heartbeat affects the quality of your lived experience. This connection between heart rhythm and emotional experience brings us full circle to the evolutionary origins of cardiac pacemaking. The same autonomic properties that allow the heart to beat without conscious control also integrated into broader physiological systems regulating stress, emotion, and social engagement. The vagus nerve, which connects the brain to the heart and other organs, evolved in vertebrates as part of a system that regulates social behavior and emotional states. This system, which the neuroscientist Stephen Porges has called the social engagement system, links heart rate regulation to facial expression, vocal tone, and other aspects of social communication. When we understand the evolutionary context, Thompson notes, we see that the automatic nature of cardiac pacemaking is not just about keeping the individual alive. It is about freeing up neural resources for the complex social behaviors that characterize vertebrate and especially mammalian life. This perspective transforms how we understand the relationship between autonomic functions like heartbeat and the conscious social aspects of human experience. They are not separate systems but deeply integrated components of a complex whole, shaped by hundreds of millions of years of evolution. The strange evolutionary trick that keeps you alive, then, is not just the automatic beating of your heart. It is the integration of this ancient, autonomous rhythm into the symphony of neural, hormonal, and psychological processes that constitute your lived experience. Your heartbeat is both the most basic prerequisite for your existence and an ongoing participant in the richness of your conscious life. As the sun rises over a quiet suburban neighborhood, Emma Williams laces up her running shoes. At 42, she has completed 3 marathons and dozens of shorter races. As she steps outside and begins her familiar route, her heart rate gradually increases from its resting 52 beats per minute to around 140 as she settles into her pace. Emma does not consciously command this cardiac acceleration. She does not need to. Her heart rate increases automatically in response to changing metabolic demands, regulated by a complex interplay between her pacemaker cells, autonomic nervous system, and circulating hormones. This seamless adaptation is the final piece of the evolutionary trick we have been exploring. Not just the automatic generation of heartbeats, but the sophisticated regulation of their timing and strength in response to changing conditions. What is truly remarkable about the cardiac pacemaking system, explains Dr. Ziegler, is not just that it works autonomously, but that it integrates so many different inputs. The sinoatrial node receives signals from the sympathetic and parasympathetic nervous systems, responds to circulating hormones like adrenaline, and adjusts to changes in blood chemistry, temperature, and stretching of the heart tissue itself. This integrative capacity allows for precise matching of cardiac output to metabolic demand, from the slow, strong beats of deep sleep to the rapid, powerful contractions during intense exercise. It represents the culmination of hundreds of millions of years of evolutionary refinement. The basic property of automaticity, the ability of pacemaker cells to generate electrical impulses without external stimulation, evolved early in animal history. But the sophisticated regulation of this automaticity to meet the demands of active, warm-blooded lifestyles required additional adaptations. Mammals and birds, which independently evolved high-energy lifestyles supported by efficient four-chambered hearts, show particularly advanced regulation of cardiac function. Their pacemaker systems integrate more variables and respond more precisely to changing conditions than those of reptiles, amphibians, or fish. This regulatory sophistication reaches its peak in humans and other primates, whose hearts must support not just physical activity but also complex social behaviors and extended periods of cognitive effort. The human heart can rapidly adjust its output in response to emotional stimuli, anticipated effort before activity even begins, and subtle social cues. Human cardiac regulation shows extraordinary sensitivity to social context, notes Dr. Thompson. Meeting a new person, speaking in public, or even thinking about an emotionally charged situation can trigger immediate changes in heart rate and rhythm. This sensitivity reflects the importance of social relationships in human evolution. These regulatory adaptations explain why emotional stress can trigger cardiac events in vulnerable individuals. The same integrative capacity that allows the heart to respond appropriately to physical demands also connects it to emotional processing systems that evolved much later. This connection between ancient cardiac regulatory systems and newer emotional processing networks creates both vulnerabilities and opportunities. Stress management techniques like meditation, deep breathing, and biofeedback can leverage this connection to improve cardiac health by modulating autonomic influences on the heart's pacemaker system. As Emma completes her run and begins her cool-down walk, her heart rate gradually returns to its resting level. This deceleration, like the earlier acceleration, occurs automatically through the interaction of multiple regulatory systems. Her breathing slows, her parasympathetic nervous system becomes more active, and the direct sympathetic inputs to her sinoatrial node decrease. This return to baseline represents another crucial aspect of the evolutionary trick keeping Emma alive, the maintenance of homeostasis, or physiological balance. The heart's regulatory systems do not just respond to changing demands but actively work to restore baseline conditions when those demands subside. Homeostatic regulation is among the most ancient and fundamental principles of physiology, present even in single-celled organisms. The sophistication of cardiac homeostasis in humans represents the culmination of billions of years of evolutionary refinement of these basic regulatory principles. Homeostasis is not just about maintaining a single, fixed state, explains Dr. McKinnon. It is about maintaining appropriate physiological conditions for the current context. The heart's regulatory systems are constantly evaluating metabolic demands, emotional states, environmental conditions, and many other variables to determine the appropriate cardiac output for each moment. This contextual regulation allows Emma to shift seamlessly from sleep to waking, from rest to exercise, from calm to excitement, all without consciously controlling her heart rate. The evolutionary trick that keeps her alive is not just the beating of her heart, but the precise tuning of that rhythm to meet her ever-changing needs. As she returns home and steps into the shower, Emma remains blissfully unaware of the ancient cellular machinery that maintained her heartbeat throughout her run. She does not need to understand the evolutionary history of HCN channels, the complex genetics of sinoatrial node development, or the intricate feedback loops regulating her heart rate. The strange evolutionary trick that keeps her alive operates below the threshold of awareness, allowing her conscious mind to focus on other matters. And this, perhaps, is the most remarkable aspect of this evolutionary adaptation: its transparency to conscious experience. Despite its fundamental importance for survival, the automatic nature of cardiac pacemaking allows it to recede from awareness, creating space for the higher cognitive functions that define human experience. There is a beautiful economy in this arrangement, reflects Dr. Simmons. The most basic life-sustaining functions are delegated to ancient, reliable autonomous systems, freeing the newer, more energetically expensive conscious brain for the complex social, cognitive, and creative activities that characterize human life. This delegation of vital functions to autonomous systems reflects a principle that extends beyond cardiac regulation to many aspects of physiology. Breathing, digestion, hormone secretion, and countless other processes occur largely without conscious control, regulated by evolutionarily ancient mechanisms that long predate human consciousness. When we appreciate this evolutionary history, Simmons continues, we gain a deeper understanding of both our robustness and our vulnerability. The autonomous systems that keep us alive are incredibly reliable. Your heart will beat billions of times without failure, but they are also susceptible to disruption when pushed beyond the conditions they evolved to handle. This perspective helps explain the modern epidemic of lifestyle-related cardiovascular disease. The cardiac regulatory systems that evolved to support intermittent high-energy activities, like hunting or escaping predators, and long periods of moderate activity, like gathering or walking, are not well adapted to the combination of chronic stress, sedentary behavior, and caloric excess that characterizes many contemporary lifestyles. Understanding the evolutionary context of cardiac function offers insights not just into disease but into potential interventions. Exercise, stress reduction, adequate sleep, and social connection all support the proper functioning of the evolved systems regulating the heart. These interventions work precisely because they align with the conditions under which these regulatory systems evolved. As Emma finishes her shower and prepares for her day as an elementary school teacher, her heart continues its steady rhythm, adapting moment by moment to her changing needs. The evolutionary trick that keeps her alive continues to operate, as it has since before her birth, as it will until the moment of her death, without her conscious input or awareness. This automatic, reliable beating, this strange evolutionary trick, is both utterly ordinary and absolutely miraculous. It is the rhythm that sustains not just Emma's life but all human life, a rhythm with origins reaching back to the dawn of animal existence, a rhythm that connects us to our deepest evolutionary past even as it enables our present and future. Each beat of your heart represents the culmination of nearly 2 billion years of evolutionary history, from the first pulsing movements of primitive multicellular organisms to the sophisticated four-chambered pump that now sustains your life. The strange evolutionary trick that keeps you alive, the automatic generation of rhythmic electrical impulses by specialized pacemaker cells, emerged not through intelligent design but through the slow, blind process of mutation, selection, and adaptation. This trick depends on a remarkable instance of evolutionary repurposing. Ion channels that originally evolved for basic cellular functions in ancient organisms were gradually adapted for new roles in coordinating the contractions of primitive hearts. Genetic regulatory networks that initially controlled the development of digestive systems in simple invertebrates were co-opted for cardiac development in more complex creatures. The sinoatrial node, that tiny, specialized region that sets the rhythm of your heart, is not just a functional component of your cardiovascular system, but a living record of evolutionary history. Its location at the junction of the right atrium and superior vena cava reflects its origins in the pacemaker region of the primitive tubular hearts of early vertebrates. Its cellular properties connect it to regulatory mechanisms present in organisms that lived hundreds of millions of years before hearts existed. This evolutionary perspective transforms how we understand not just cardiac function but human existence itself. It reveals that our lives depend on ancient, autonomous systems operating largely outside conscious awareness or control. It highlights the deep interconnections between physical processes like heartbeat and experiential aspects of life like emotion and social connection. It reminds us that we are not separate from nature but embodiments of its long evolutionary history. The next time you place your hand on your chest and feel your heartbeat, remember that you are experiencing not just a mechanical process but a living connection to your evolutionary past. Each beat echoes the rhythmic contractions of countless ancestors stretching back to the dawn of animal life. Each pause between beats represents the precise timing established by ion channels with ancient origins. Each acceleration or deceleration in response to activity or emotion reflects regulatory systems refined over hundreds of millions of years. This strange evolutionary trick, the automatic, reliable generation of heartbeats, is both utterly ordinary in its ubiquity and absolutely extraordinary in its complexity and importance. It operates continuously from before your birth until the moment of your death, adapting to changing conditions, responding to physical and emotional states, maintaining the rhythm that sustains your life. And perhaps most remarkably, it does all this without your conscious input, without your awareness, without your control, allowing your conscious mind to focus on the uniquely human activities that define your experience. Learning, creating, connecting, loving. The strange evolutionary trick that keeps you alive is not just a biological curiosity but a foundational aspect of what it means to be human. To be a creature whose basic existence is maintained by ancient autonomous systems while consciousness explores the vast possibilities of art, science, relationship, and meaning. Your heart beats so that you can live, not just in the basic biological sense but in the full, rich sense that encompasses all aspects of human experience. This is the ultimate significance of the strange evolutionary trick that keeps you alive. It creates the physiological stability that allows for the emergence of consciousness, creativity, and connection. In the steady rhythm of your beating heart, biology and biography converge, and the vast sweep of evolutionary history meets the intimate reality of your individual existence.